Transplantation, graft or transplant, transfer of living the cells, tissues and organs from one part of the body to another part or from one individual to the another. Why we are going to transplantation? So many needs in humans. If there will be a damaged organs and if there is non-functional organs is present. For the definition of a transplantation, the implantation of the non-self tissue into the body, that is, the process of taking cells, tissues or organs called the graft transplant from one part or individual and placing them into the another, usually the different individual. Donor, the individual who provides the graft. The recipient or the host, the individual who receives the graft. Types of transplantation: autograft, isograft, allograft and xenograft. Autograft is nothing but within the individual and xenograft is between the species who are non-identical. Autograft, it is a tissue or organ taken from one side and engrafted to the another side in the same individual. Example, skin grafting in burns and the destructive injuries. Isograft, it is a graft taken from an individual and placed on another individual of the same genetic constitution. Example, graft between identical twins. Allograft, they are formerly called homograft. It is a graft between the two genetically non-identical members of the same species. Example, kidney transplant from one person to any other except an identical twins. Xenograft, formerly called heterograft. These are the graft between the members of the different species and are called xenograft. Example, from monkey to humans. Autograft and isograft are usually accepted and survive. The allograft and the xenograft are usually rejected due to the genetic and the antigenic incompatibility. The allograft reaction, rejection of the graft by the recipient is called the allograft reaction. The graft becomes vascularized during the first 2 or 3 days and appears to be accepted initially. By 4th day, the inflammation occurs and the graft is invaded by the lymphocytes and macrophages. Vascularity diminishes due to the thrombosis and the graft undergoes the ischemic necrosis. By the 10th day, graft sloughs off. This event resulting in the rejection of the allograft is known as the first set of response. That is, the cell-mediated reaction is responsible for the reaction. Rejection is primarily by the helper T lymphocyte, which activate the cytotoxic T lymphocyte, macrophages and the B lymphocyte. When a second allograft from the same donor is applied on the sensitized recipient, that is, in which the previous graft has been rejected by the first set response, it will be rejected in the accelerated fashion. Necrosis sets nearly and graft slough off by the 6th day. This type of rejection is known as second set response. Antibodies play a dominant role along with the cell-mediated immunity. Histocompatibility testing. Procedure used for matching the donor and the recipient for the transplantation are as follows. ABO grouping, tissue typing, cytotoxicity test, mixed leukocyte reaction, screening for the presence of preformed antibodies to the allogenic HLA cross-matching. Prevention of the graft rejection. One is immunosuppression. The immunosuppression is produced to the recipient so that the transplanted tissue survives for a longer period. Radiation, corticosteroid and the anti-lymphocytic serum, they are employed for the immunosuppression in the clinical transplantation. Fungal metabolites such as cyclosporin A and the rapamycin, they are also used. They show the specific T cell inhibitory activity. Transplantation in anatomically protected sites. There are certain privileged sites where the allograft are permitted to survive. This included the cornea, cartilage and the testicles grafting. Lack of vascularity prevents the graft rejection in of the corneal transplantation. Graft versus host reaction. Graft rejection is due to the reaction of the host to the grafted tissue. The contrary situation in which the grafted tissue may react to and reject the host. This is known as graft versus host reaction. The graft versus host reaction occurs when the following conditions are present. Graft like bone marrow, lymphoid tissue, splenic tissue, etc. contains the immunocompetent T cell. The recipient possesses the transplantation antigen that are absent in the graft. The recipient may not reject the graft The host immunological responsiveness must be either destroyed or so impaired following the whole body irradiation, and it cannot reject a graft, that is allograft or xenograft. Tumor immunity. Tumor cells that continue to replicate fail to differentiate into the specialized tissue and become immortal. Malignant, a tumor that grows indefinitely and spreads, also called the cancer, which kills the host. Meningia, a tumor that is not capable of metastasis, that is, they cannot spread all over and does not kill the host. Carcinoma, arising from the epithelial tissue such as glands, breast, skin, and the linings of the urogenital, digestive, and the respiratory system. Sarcoma, that is a solid tumor of muscles, bone, and the cartilage that arise from the embryological mesoderm. Leukemia, disease of the bone marrow causing the excessive production of leukocytes. Lymphoma or myeloma, disease of the lymph nodes and the spleen that causes the excessive production of lymphocytes. Etiology of cancer. There may be genetic factors like mutation, translocation, and amplification. While speaking about the environmental factors, there may be UV, that is ultraviolet rays, chemicals, viral infections, the conversion of the proto-oncogen potential for the cell transformation to the oncogen cell transformation from one cell to another cell, alternation in the tumor suppressor genes. Tumor antigen. When a cell undergoes a malignant transformation, it acquires a new surface antigen and may lose some normal antigen. A malignant tumor antigenetically different from the normal tissues of the host, it is considered as an allograft and expected to induce the immune response. The antigens that are present in the tumor cells but are absent in the corresponding normal cell of the host are known as tumor antigen. Two types: tumor-specific antigen and tumor-associated antigen. Tumor-specific antigen, these are the antigen present on the membranes of the malignant cells and induce an immune response when the tumor is transplanted in the syngeneic animals. Such tumor-specific antigen reject the tumor transplant in the immunized host. These are termed as tumor-associated transplantation antigen, tumor-specific transplantation antigen. Tumor-associated antigen. These are the fetal antigen that are present in the embryonic and the malignant cell but not in the adult normal cell. Example, alpha fetoprotein in the hepatoma and the carcinoma embryonic antigen in the colonic cancer. Immune response in the malignancy. T-cell mediated immunity. Lymphokines results which helps in the destruction of the tumor cells. Humoral response, this is a beneficial one. Immunotherapy of a cancer. Non-specific active immunotherapy employs a BCG vaccine. This evokes the tumor immunity, enhancing the macrophage cytotoxicity. Ivamycin. While speaking about the specific immunotherapy, the monoclonal antibodies to tumor antigens. Lymphokines activated killer cells obtained by the treatment of a natural killer cells with the interleukin used in the renal carcinoma. Thank you.